Hey everyone. Today we're diving into the fascinating world of charophytes. These are special freshwater green algae that have captured the attention of scientists worldwide. Let's start by taking a closer look at what charophytes actually look like under a microscope. Here we can see the cellular structure of these remarkable organisms. Charophytes are a distinct group of freshwater green algae. They live in clean, freshwater environments and have some very special characteristics that make them unique. But here's what makes charophytes truly special. Scientists believe they are the closest living relatives to land plants. This makes them incredibly important for understanding how plants evolved from water to land. Charophytes come in many different forms. Scientists have identified several major groups, from early divergent types to more advanced forms that are even closer to land plants. Understanding charophytes helps us piece together one of the most important events in Earth's history: how life moved from water to land. By studying these algae, we can learn about the evolutionary steps that led to all the plants we see around us today. So remember, charophytes are special freshwater green algae that serve as a living bridge between aquatic and terrestrial life. They're our best window into understanding how plants conquered the land billions of years ago. What makes charophytes so special in the plant kingdom? The answer lies in their remarkably close relationship with land plants. Charophytes are actually the closest living relatives to all terrestrial plants, making them incredibly important for understanding plant evolution. The first clue to their close relationship comes from examining their cellular structures. When we look inside charophyte cells and compare them to land plant cells, we find remarkable similarities. Here we can see a charophyte cell from Spirodra on the left and a typical land plant cell on the right. Notice how both have similar organelles: chloroplasts for photosynthesis, a nucleus for genetic control, and cell walls for structure. Both cell types share cellulose cell walls, chloroplasts that store starch, similar flagellated reproductive cells, and they both use a structure called a phragmoplast during cell division. These aren't just coincidences, they're evidence of shared evolutionary history. To truly understand this relationship, we need to look at the evolutionary family tree. Just like human families, plants have evolutionary relationships that can be traced through their shared characteristics. This phylogenetic tree shows the evolutionary relationships between charophytes and land plants. Notice how charophytes branch off just before land plants evolved. This makes them our best living example of what the ancestors of land plants might have looked like. The similarities don't stop at cellular structure. Charophytes and land plants also share similar reproductive strategies, including the formation of specialized reproductive structures and complex life cycles. Both groups show complex sexual reproduction, alternation between different life cycle stages, protected reproductive structures, and similar ways of forming reproductive cells called gametes. Understanding this close relationship between charophytes and land plants is crucial for scientists studying plant evolution. Charophytes give us a window into the past, showing us what the transition from aquatic to terrestrial life might have looked like. Charophytes act like living fossils, helping us understand how plants evolved from aquatic ancestors to colonize land. They reveal ancestral characteristics and show us the evolutionary innovations that made terrestrial plant life possible. In essence, charophytes represent the bridge between aquatic and terrestrial plant life, making them invaluable for understanding one of the most important transitions in the history of life on Earth. Charophytes have a fascinating global distribution. These remarkable algae can be found all around the world, but they have very specific preferences for where they like to live. Charophytes are truly cosmopolitan organisms. You can find them on every continent except Antarctica, thriving in both temperate and tropical regions across the northern and southern hemispheres. Charophytes are primarily freshwater organisms with very specific habitat preferences. They thrive in clean lakes, quiet ponds, slow-moving streams, and occasionally in brackish water environments where freshwater meets saltwater. When it comes to where they grow, charophytes prefer shallow areas with soft mud bottoms, though they can also attach to sandy or rocky surfaces. They typically live in the sunlight penetration zone, usually between half a meter to 10 meters deep. Here's something really important about charophytes. They are extremely sensitive to water quality. They require clean water with low nutrient levels and high clarity. This sensitivity makes them excellent indicators of environmental health, but it also means they quickly disappear from polluted areas. When you look at charophytes under a microscope, you can see their distinctive crescent-shaped cells with visible green chloroplasts. In their natural habitat, they often form underwater meadows that provide important shelter for fish and help maintain water clarity. The key takeaway is that charophytes are cosmopolitan organisms found worldwide in clean freshwater environments. They prefer shallow areas with soft substrates, but are highly sensitive to pollution, which makes them excellent indicators of environmental health. Charophytes have a fascinating and unique structure that sets them apart from other algae. Their body plan is surprisingly complex for aquatic organisms. The charophyte body consists of a long, slender, jointed main axis. This axis is divided into distinct sections called nodes and internodes. At each node, charophytes develop whorls of lateral branches. These branches extend outward in a circular pattern like spokes on a wheel. Think of this structure like a plant stem, with leaves arranged in circles around it. This arrangement is called a whorl pattern. On the left, we see a plant stem with leaves in circular arrangements. On the right, we see the similar pattern in charophytes with their lateral branches. Now let's look at the cellular level to understand how this structure is built. Here we can see the internal organization of charophyte cells. This diagram shows a Spirogyra cell, which is related to charophytes. Notice the spiral chloroplast, cell wall, and other structures that support the overall body plan. This unique node and internode structure with whorled branches provides several advantages in aquatic environments. The branch structure increases surface area for photosynthesis, improves nutrient absorption, provides stability in water currents, and allows efficient light capture from all directions. This sophisticated structural organization demonstrates how charophytes have evolved to thrive in their aquatic habitats while maintaining the complex body plan that links them to land plants. Charophyte cells have a unique internal structure that sets them apart from many other algae. The most distinctive feature is that they are coenocytic, meaning each cell contains multiple nuclei and numerous chloroplasts. This coenocytic structure means that unlike typical plant cells with single nuclei, charophyte cells function as multinucleated units, allowing for more complex cellular processes and larger cell sizes. Let's take a closer look at the chloroplasts, which are the powerhouses of photosynthesis in these cells. Charophyte chloroplasts have a complex internal structure with thylakoids organized into stacks called grana, surrounded by the stroma. This organization is very similar to what we see in land plants. Now let's examine the cell wall, which provides structure and protection to charophyte cells. Charophyte cell walls contain the same major components found in land plant cell walls: cellulose for strength, xyloglucans for flexibility, pectins for binding, and various proteins for specialized functions. This remarkable similarity in cell wall composition provides strong evidence for the close evolutionary relationship between charophytes and land plants. It suggests they share a common ancestor and similar cellular mechanisms. The cellular structure of charophytes, with their coenocytic organization and land plant-like cell walls, represents a crucial evolutionary bridge between simple algae and complex terrestrial plants. Charophytes have developed several clever ways to reproduce without needing a partner. These asexual reproduction methods allow them to create exact copies of themselves quickly and efficiently. The first method is fragmentation. Think of it like breaking a twig from a tree. When a piece of the charophyte breaks off, it can grow into a completely new plant. The second method involves amylum stars. These are special starch-filled structures that can develop into new plants. They're like tiny energy packages that contain everything needed to start a new charophyte. The third method is bulbal formation. Bulbils are small, specialized structures that grow on the parent plant and can detach to form new individuals. This image shows how bulbils develop and eventually separate to create new plants. These asexual reproduction methods give charophytes a major advantage. When conditions are favorable, they can rapidly multiply and spread throughout their habitat without waiting to find a mate. This allows them to quickly colonize new areas and take advantage of good growing conditions. Charophytes have a fascinating approach to sexual reproduction that involves specialized structures and a unique cellular process. Let's explore how these algae create the next generation. Sexual reproduction in charophytes involves two specialized structures. The antheridia are the male reproductive organs containing sperm cells. The eugonia are the female reproductive organs containing egg cells. Most charophyte cells are haploid, meaning they contain only one set of chromosomes. However, during sexual reproduction, something special happens. A diploid zygote is formed with two sets of chromosomes. During fertilization, a sperm cell from the antheridia swims to and fuses with an egg cell in the eugonia. This fusion creates a diploid zygote, the only diploid stage in the charophyte life cycle. The result is a diploid zygote containing genetic material from both parents. This zygote represents the brief diploid phase in the charophyte reproductive cycle. The diploid zygote doesn't stay diploid for long. It immediately undergoes meiosis, a special type of cell division that reduces the chromosome number by half. This process produces 4 new haploid cells, ready to grow into new charophyte organisms. This creates a beautiful cycle. Haploid charophyte cells produce gametes, fertilization creates a diploid zygote, and meiosis returns to the haploid state. It's like nature's way of shuffling genetic material while maintaining the predominantly haploid lifestyle of charophytes. This sexual reproduction strategy allows charophytes to combine genetic material from different individuals, creating genetic diversity that helps them adapt to changing environments while maintaining their efficient haploid cellular organization. Charophytes have a fascinating two-stage life cycle that alternates between different cellular states. Understanding this cycle helps us see how these algae ensure their survival and genetic diversity. The charophyte life cycle consists of 2 distinct stages. The haploid stage, where cells contain one set of chromosomes, is the predominant and longest-lasting stage. This is where charophytes develop their reproductive structures. The diploid stage, where cells contain 2 sets of chromosomes, is much briefer. This stage exists only as a zygote, the fertilized cell that forms when reproductive cells combine. The key process in the charophyte life cycle is meiosis. When the diploid zygote undergoes meiosis, it divides to create 4 new haploid cells. This process is crucial for genetic diversity. Each of the 4 haploid cells produced by meiosis develops into a mature gametophyte. These gametophytes will eventually produce the reproductive structures needed to continue the cycle. This 2-stage life cycle provides charophytes with important advantages. The meiosis process creates genetic diversity, giving populations the variation needed to adapt to changing environments. This ensures the long-term survival of charophyte species. In summary, the charophyte life cycle, with its 2 distinct stages, the predominant haploid gametophyte and the brief diploid zygote, ensures both successful reproduction and the genetic diversity necessary for species survival. When environmental conditions become harsh, charophytes have developed remarkable survival strategies. They can enter a state of hibernation to wait out difficult periods and emerge when conditions improve. Charophytes face many environmental challenges, including extreme cold, drought, and excessive heat. Rather than dying, they have evolved 3 distinct hibernation methods to survive these tough times. The first hibernation method involves the charophyte remaining as a green plant but entering a dormant state. The plant reduces its metabolic activity and growth while maintaining its basic structure, waiting for favorable conditions to return. The second method involves producing bulbils, specialized reproductive structures that can survive harsh conditions. These small, nutrient-rich structures detach from the parent plant and remain dormant until environmental conditions improve. The third hibernation method involves sexual reproduction to form oospores. These are fertilized eggs surrounded by a thick, protective wall that can withstand extreme conditions for extended periods. The gametangia shown here produce the gametes that fuse to create these hardy oospores. The choice of hibernation method depends on environmental conditions like temperature, water availability, nutrients, and light. When favorable conditions return, charophytes can quickly resume active growth, making them remarkably resilient organisms that can thrive across changing environments. This hibernation ability is a key reason why charophytes have been so successful evolutionarily, allowing them to colonize diverse freshwater habitats worldwide and survive through millions of years of environmental change. Understanding how charophytes are classified helps us see their relationships to other organisms and to each other. Taxonomy is the science of naming and organizing living things into groups based on their similarities and evolutionary history. Taxonomy is like organizing a huge library. Just as books are grouped by subject and author, scientists group organisms by their characteristics and evolutionary relationships. Charophyte classification has changed dramatically over time. Scientists once relied mainly on what organisms looked like under a microscope. Today, we use DNA analysis to understand true evolutionary relationships. Molecular data analysis has been crucial in understanding charophyte relationships. By comparing DNA sequences, scientists discovered that some organisms that look similar are actually distantly related, while others that look different share recent common ancestors. Today, charophytes are organized into several main classes. Each class represents a distinct evolutionary lineage with unique characteristics. Charophyceae includes the most complex forms, while Zygnomatophyceae are considered the closest relatives to land plants. This phylogenetic tree shows the evolutionary relationships between different charophyte classes and their connection to land plants. Notice how some charophyte groups are more closely related to land plants than others, revealing the step-by-step evolution from aquatic algae to terrestrial plants. The key takeaway is that modern charophyte classification represents a revolution in our understanding. By combining molecular data with traditional observations, scientists have revealed the true evolutionary story of these important algae and their relationship to all land plants. Charophyte genomics represents one of the most exciting frontiers in understanding plant evolution. Scientists around the world are now sequencing the complete genomes of these remarkable algae. Multiple genome sequencing projects are currently underway, focusing on different charophyte species. These projects use advanced DNA sequencing technology to read every letter of their genetic code. These genomic studies are revealing detailed evolutionary relationships between charophytes and land plants. The genetic data confirms that charophytes are indeed the closest living relatives to all land plants. By comparing charophyte genomes with those of land plants, scientists can identify which genes were crucial for the transition from aquatic to terrestrial life millions of years ago. Comparative genomic analysis between charophytes and land plants is uncovering the genetic toolkit that enabled plants to colonize land. This includes genes for cell wall formation, stress tolerance, and developmental control. These genomic studies are revealing specific gene families involved in cell wall biosynthesis, stress responses, reproductive mechanisms, and developmental pathways that were essential for plant evolution. Charophyte genomics is like having a key that unlocks the secrets of plant evolution. These genetic blueprints help us understand how plants developed the innovations needed to thrive on land. As more charophyte genomes are sequenced and analyzed, we gain deeper insights into evolutionary innovations, gene functions, and the remarkable journey that led to the diversity of plant life we see today. Charophytes are remarkable organisms that help scientists understand how plants develop stress tolerance. When early plants made the dramatic transition from water to land millions of years ago, they faced entirely new environmental challenges. Environmental stress comes in many forms. Desiccation, or drying out, was perhaps the biggest challenge. Plants also had to cope with freezing temperatures, intense sunlight, and harmful ultraviolet radiation. These stresses could damage or kill plant cells. Charophytes developed sophisticated responses to these stresses. When faced with environmental challenges, they activate complex signaling pathways. This includes phytohormone signaling that triggers protective mechanisms like cell wall strengthening, mucilage production, and spore formation. At the cellular level, Charophytes show remarkable adaptations. Their cell walls become reinforced, chloroplasts develop protective mechanisms, and the cytoplasm adjusts its composition to maintain function under stress. These cellular changes help us understand how plant cells evolved to survive on land. Ecophysiological studies of charophytes provide valuable data for understanding plant adaptation. Scientists study how these organisms respond to controlled stress conditions, measuring cellular changes and survival rates. This research helps us understand both evolution and modern plant stress responses. Understanding stress tolerance in charophytes gives us insights into one of the most important transitions in Earth's history. These remarkable organisms continue to inform our knowledge of plant biology, evolution, and how life adapts to environmental challenges. Charophytes serve as excellent biomonitors, acting like natural water quality detectors in aquatic ecosystems. Biomonitors are living organisms that scientists use to assess environmental health. They act like natural sensors, responding to changes in their surroundings. Charophytes work like the famous canaries that miners once used in coal mines. These birds would stop singing if dangerous gases were present, warning miners of unsafe conditions. When charophytes are thriving in an aquatic environment, it's a strong indicator of good water quality. Their presence tells us the water is clean, with low pollution levels and a healthy ecosystem. However, when charophytes are absent or dying, it signals environmental problems. Their disappearance warns us of poor water quality, high pollution levels, and ecosystem stress. Charophytes are particularly sensitive because they require very specific water conditions to survive. They react quickly to chemical changes and cannot tolerate contamination, making them excellent early warning systems. The key takeaway is that charophytes function as nature's water quality sensors. Their presence or absence provides valuable information about the health of aquatic ecosystems, making them indispensable tools for environmental monitoring. Charophytes face serious conservation challenges due to their extreme sensitivity to environmental changes. Understanding why these algae are endangered helps us appreciate the urgent need for protection efforts. Charophytes are extremely sensitive to water quality changes. Unlike many other aquatic plants that can tolerate pollution, charophytes require very clean, stable conditions to survive and reproduce. Multiple environmental threats put charophytes at risk. Pollution from drainage systems, agricultural runoff, and urban development creates conditions that charophytes simply cannot tolerate. This image shows how human activities directly impact their aquatic habitats. Conserving charophytes is crucial for multiple reasons. They represent our closest link to understanding plant evolution, serve as early warning systems for ecosystem health, contribute to biodiversity, and provide invaluable opportunities for scientific research. When charophytes thrive, they create beautiful underwater meadows like this. These healthy populations require pristine water conditions with specific characteristics that support their growth and reproduction. Effective charophyte conservation requires a multifaceted approach. We must protect water quality, preserve and restore their habitats, and conduct ongoing research to monitor populations and understand threats. The key to charophyte conservation is maintaining clean water. When we protect water quality for charophytes, we're also protecting entire aquatic ecosystems. Every action we take to reduce pollution and preserve clean water helps ensure these remarkable algae can continue to thrive for future generations. Science never stops. Even today, researchers around the world are discovering new charophyte species. These discoveries help us understand the incredible diversity of these algae and their important roles in aquatic ecosystems. Discovering new charophyte species involves several careful steps. Scientists start by collecting samples from freshwater habitats like swamps, lakes, and streams. They look for algae that might be different from known species. Once collected, scientists examine the specimens under powerful microscopes, analyze their DNA, and compare them with existing species. If they find significant differences, they can describe a new species. Each new charophyte species we discover adds another piece to the puzzle of algal diversity. Charophytes are divided into 6 major groups: and new species help us understand the unique characteristics of each group. These discoveries also help scientists trace the evolutionary history of charophytes and understand how they are related to land plants. Every new species provides valuable information about adaptation and evolution. New charophyte discoveries have far-reaching impacts on science. They help us understand ecosystem relationships, provide insights into evolution, guide conservation efforts, and expand our overall scientific knowledge. The phylogenetic tree shows how new species fit into the broader picture of plant evolution. Each discovery helps scientists refine our understanding of how life on Earth has evolved and diversified. Remember these key points about charophyte discoveries. Science is always evolving as we find new species. Each discovery adds valuable information to our understanding. These findings help with conservation efforts and future research. And most importantly, there's always more to learn about the fascinating world of charophytes. The world of charophytes continues to surprise us with new discoveries, reminding us that nature still holds many secrets waiting to be uncovered. Let's examine a specific example of charophyte discovery, Charophyta. This fascinating species demonstrates how scientists continue to find and classify new charophytes in the modern era. This specimen was discovered in freshwater environments in China. Here, we can see the actual pressed specimen that scientists studied to identify and describe this new species. Scientists identified this species using two complementary approaches: morphological evidence and molecular evidence. Let's explore what each of these methods involves. Morphological evidence involves studying the physical structure and appearance of the organism. Scientists examine the branching patterns, cell arrangements, and reproductive structures under microscopes. Molecular evidence involves analyzing the organism's DNA sequences. Scientists compared genetic material to distinguish this species from closely related charophytes and confirm it as a new species. The discovery of Charophyta demonstrates that charophyte research is very much alive. Scientists continue to find new species, expanding our understanding of charophyte diversity and evolution. This example shows how combining traditional morphological studies with modern molecular techniques allows scientists to accurately identify and classify new charophyte species, contributing to our growing knowledge of these important algae. Charophyte meadows are some of the most valuable underwater ecosystems on Earth. These dense communities of charophyte algae create underwater forests that support entire aquatic food webs. Charophyte meadows are dense underwater communities that form in shallow bays and lakes. These algae grow on soft, muddy bottoms, creating vast underwater forests that can cover entire lake floors. One of the most important functions of charophyte meadows is improving water clarity. These algae act like natural water filters, removing sediments and excess nutrients that would otherwise make water murky and promote harmful algae blooms. Charophyte meadows provide critical habitat for a wide variety of aquatic animals. Small fish use these underwater forests as nurseries, while invertebrates find food and shelter among the dense vegetation. The meadows also serve as important spawning grounds and feeding areas for many species. Unfortunately, charophyte meadows are under threat from pollution, nutrient runoff, and habitat destruction. These sensitive ecosystems are among the first to disappear when water quality declines. Protecting them requires monitoring water quality, reducing nutrient inputs, and preserving shoreline habitats. Charophyte meadows represent some of nature's most valuable underwater habitats. By understanding their importance and taking action to protect them, we can help preserve these critical ecosystems for future generations. Fossil charophytes are like ancient time capsules, preserved in rock layers. These microscopic remains help scientists unlock secrets about Earth's past and understand how life evolved over millions of years. Fossil charophytes are the preserved remains of these ancient algae found in sedimentary rocks. When charophytes died millions of years ago, their structures became buried and fossilized, creating a permanent record of their existence. Scientists use fossil charophytes for biostratigraphy, a method of dating and correlating rock layers. Different charophyte species lived during specific time periods, so finding their fossils tells us the age of the rock layer they're found in. In this geological column, we can see different rock layers. Charophyte fossils found in upper layers are younger, while those in deeper layers are older. This creates a timeline of charophyte evolution. Fossil charophytes help us understand evolutionary history by showing us how plants evolved over time. This phylogenetic tree shows the relationships between charophytes and land plants, with fossil evidence supporting these connections. Fossil charophytes truly are time capsules from the past. They preserve information about ancient climates, environments, and evolutionary processes that occurred millions of years ago, giving us invaluable insights into Earth's history and the development of plant life. When we look at charophyte diversity, the numbers tell an interesting story. Scientists have identified approximately 294 different species of charophytes worldwide. These 294 species are organized into just 7 genera, showing how taxonomists group related species together based on their shared characteristics. To visualize this diversity, imagine each genus as a family group. Some genera like Chara contain many species, while others like Nidolopsis have fewer representatives. Here we can see what charophytes actually look like in their natural aquatic environment. Each of the 294 species plays a unique role in its ecosystem. Some species thrive in shallow, warm waters, while others prefer deeper, cooler environments. Some can tolerate brackish water, while others need pure freshwater conditions. This diversity means 294 different approaches to survival, reproduction, and ecosystem interaction. Each species has evolved its own unique life strategy over millions of years. So when we say there are 294 charophyte species in 7 genera, we're talking about a remarkable diversity of life forms, each perfectly adapted to its specific aquatic environment and ecological role. Charophytes face a critical challenge in today's changing world. These remarkable algae are considered among the most sensitive and threatened plant groups on our planet. Several factors make charophytes particularly vulnerable to environmental threats. They are extremely sensitive to water quality, cannot tolerate pollution, require specific habitats, and are vulnerable to climate changes. Pollution from industrial and urban sources poses a major threat to charophyte habitats. Even small amounts of chemical runoff can devastate entire populations of these sensitive organisms. The threats facing charophytes are severe and widespread. Many species have already gone extinct in polluted waters, habitat loss is accelerating, and climate change is altering water chemistry. Once populations decline, recovery is extremely slow. Conservation efforts must begin immediately to protect charophytes. We need to protect existing habitats from pollution, restore degraded ecosystems, monitor water quality regularly, and educate communities about their critical importance. Every charophyte specimen represents millions of years of evolution and plays a crucial role in freshwater ecosystems. These preserved specimens remind us of the incredible diversity we stand to lose if we don't act now. Time is running out for many charophyte species. As one of the most threatened plant groups on Earth, they need our immediate protection. The actions we take today will determine whether future generations can study and benefit from these remarkable organisms. Understanding charophyte communities requires a comprehensive research approach. Scientists use 4 main methods that work together to give us the complete picture of these fascinating organisms. The first method is morphological analysis. Scientists examine the physical structure and form of charophytes, their nodes, internodes, and cellular architecture. This tells us how they're built and how they function. Ecological methods study how charophytes interact with their environment. This includes their habitat preferences, water quality requirements, and relationships with other organisms in their aquatic ecosystems. Molecular techniques analyze the DNA and genetic makeup of charophytes. This reveals information about their biochemistry, gene expression, and helps identify species that look similar, but are genetically different. Phylogenetic analysis studies evolutionary relationships between different charophyte species and their connection to land plants. This helps us understand how these organisms evolved over millions of years. The power comes from integrating all 4 approaches. Each method provides different pieces of the puzzle, and when combined, they give scientists a complete understanding of charophyte communities. This holistic research approach ensures that scientists get the most complete picture possible. By combining morphological, ecological, molecular, and phylogenetic methods, researchers can fully understand the complex biology of charophyte communities. After exploring charophytes throughout this series, let's summarize the key reasons why these remarkable algae matter so much to science and our planet. These are the charophyte cells we've been studying, simple yet sophisticated algae that hold keys to understanding life on Earth. First, charophytes serve as an evolutionary bridge to land plants. As the closest living relatives to all terrestrial plants, they provide crucial insights into how life made the monumental transition from aquatic to terrestrial environments. Second, charophytes act as environmental health indicators. Their extreme sensitivity to water quality changes makes them excellent biomonitors, helping scientists assess the health of aquatic ecosystems and detect pollution early. Third, charophytes serve as valuable research models while requiring urgent conservation attention. They advance our understanding of fundamental plant biology processes, yet many species are threatened by environmental changes, making their protection essential for preserving biodiversity. The bigger picture is clear. By studying and protecting charophytes, we gain valuable insights into the evolutionary history of life on Earth, learn how organisms adapt to environmental challenges, develop methods for monitoring ecosystem health, and create strategies for conserving biodiversity. These seemingly small algae connect us to our planet's past and help secure its future. Understanding charophytes isn't just about studying algae, it's about understanding the fundamental processes that shape life on our planet and ensuring we protect the delicate ecosystems that sustain us all.